Okay, looks like we are live at Club Standing for Truth with that music. Hey, Matt, if that doesn't pump you up, I don't know what will.
That's right.
We party hard, especially because it's, after 3 in the morning, but our faithful listeners are still here with us. We've already got super stickers, we've got super chats. Doki Doki Bible Club, we love you. Thank you so much, guys, for the support. As you know, we want to put out as much content, as much videos, debates, write as many books as we can. And that can only be possible because of all our amazing support, all our amazing viewers. So you guys are awesome. We appreciate it. We thank you so much. This is going to be fun. I'm looking forward to this one. I do want to say, guys, if you haven't yet, watch the interview Demolishing Abiogenesis and Origin of Life with Dr. Ryan Hayes. Please check it out. That was yesterday, getting a lot of good feedback. I don't expect any real rebuttals from the old atheists. And also September 24th, be here. We've got Kent Hovind, Dr. Dino himself. He is going to be debating Derek Barnes on his evolution in science. That's going to be a lot of fun too. And we should have Dr. Hayes on a couple more times. We were discussing some lectures he'd like to give us, and I'm looking forward to it. It's going to be awesome. Lucas says 9:22 a.m. No problem here. Awesome. So yeah, let's have some fun here. We're going to be discussing Y chromosome, a couple other things. We're going to be debunking some of our favorite critics. Mainly our most favorite currently, Gutsick Gibbon. We've got, you can look in the description box for all the videos we've made. Look at the playlists. We've addressed all her arguments thoroughly. There's a few things she said in comment sections that we're just dying to address. They're definitely running out of arguments and data. And Matt, you probably feel the same way. It's getting to the point we're just answering the same objections, answering the same questions over and over again.
Yeah, but I mean, it's okay because it gives a opportunity for other people to hear how we answer. And
exactly.
That's good, I guess.
Yeah, we've had a good back and forth in the comment section. Now, check out our last couple videos where we've gone into incredible detail on like orphan genes, DNA function. Our last video, we went into incredible detail on molecular clocks, for example, in the mitochondrial DNA. So we're not just going to rehash all of that. Therefore, if Erica is planning on responding, she's definitely going to want to respond to the arguments and evidence put forth in those videos, especially the last video. George Bond, $20 super chat. He says Friday is pizza day. George, because of you, I've been addicted again. I had pizza last night, unfortunately, almost the entire thing to myself. I had to work out for an extra hour today just to burn off those calories. Matt, I would have invited you over, brother, but you're only about 20 hours away, so you wouldn't have got here in time.
Never would have made it. Plus, I couldn't have eaten it anyway.
No, you don't want to eat that garbage. Okay, yeah, let's get right into it, Matt. I know a spur of the moment kind of comment on the endogenous retroviruses. Did you want to touch on that, brother? Obviously, we're not going to go into crazy details, guys. In 2 videos ago, our orphan gene videos, we showed a number of papers going over many of the amazing functions found in these retrotransposons. But here we're just going to go over an objection real quick. And Matt, if you want to go ahead, brother.
Yeah, I didn't want to straw man or anything that she's saying, but I guess she's making the comment that they're pretty much like junk DNA, and ERVs are non-functional, or she said some do have function, and she agrees with that, but the vast majority do not. And we're saying that the opposite is true, kind of like junk DNA. We're saying that the majority should have function, not all of them, of course, because they break in time. All ERVs have deleterious mutations and lethal deleterious mutations. So of course, not all ERVs are going to have function simply based on that fact alone. They're degrading like other things are degrading and breaking. But, you know, when we look at HERVKs, which are complete endogenous retroviruses and actually very large, we find that they all have function, but it's the little ones that are broken up into bits and pieces, the other 98,000 that are throughout the body, that, yeah, we expect that some of them will have function, of course, like probably even half of those have function because, for one thing, we know that they must be doing something because the body is supplying ATP to them. And the body doesn't waste energy giving it to something that's useless and junk. So obviously, even though we might not know what the function is, it clearly has some sort of function, or the body would not waste vital energy supplying it to these supposedly dead, worthless junk viruses that are left over. And another thing is that, you know, if these are degrading, this shows our model. It is the genetic entropy model, and that is what we're seeing in all of these. That's why they all have deleterious mutations, and some of them are completely lethal in the very large ones that are still left. So yeah, I would say that we are finding function, and remember, they're not testing these things for function. So that the fact that we're actually do find critical functions in these things does show that they are very important to some extent. Now, what the other little smaller bits and pieces actually do, besides like transcription, we don't really know yet, but we'll get there.
And what's funny is, how long have we been asking for any empirical evidence, any technical paper that actually demonstrates and not just infers or assumes these endogenous retroviruses going from non-functional to functional? They never predicted this. And Erica keeps saying, oh, one or two. There's so much more. And for sake of time, I'm not going to screen share all the numbers of papers. I did that in previous videos. So she can go look at that. But how long have we been asking for that evidence, for that specific paper there, Matt?
I would say that I really heard you start asking for that in the cats debate. You before that, though, of course, but that's when you really were just like, I just want to put it out there to everybody, including you. Just give me even a single paper. Find one if you can, because he said that was the best evidence for evolution, and he couldn't supply you anything.
So. Right. Well, what's funny too is when it comes to the retrotransposons, even the ALUs, ERVs, like I said, they've never predicted function. I've always asked the question, are these really the ancient remnants of viral infections, or are they created units of DNA function? Well, we've clearly demonstrated the function aspect. The other question is, based on what we know on the structure, for example, of retroviruses, the question is, did they originate from the outside or from the inside? Before I even answer that question, it's funny that they could assume viral DNA could be inserted randomly into the genomes of biological organisms and actually lead to something incredibly functional in gene expression, critical functions that we discuss over and over again. But if that were to happen in an evolutionary scenario, it would more than likely, disrupt the existing genes. And I always make this analogy. If you had a blindfolded painter, okay, he's obviously going to mess up the painting he is working on if he were to just start applying random brushstrokes to it. So it makes no sense that these ERVs would gain such incredible functional roles in our genome. And we've discussed how they're important in the gastrointestinal tract, cell stress responses. But here's the thing. Retroviruses, what we would say according to our model, they would have originated from within the host genome instead of invading it from the outside. And the funny thing is, this had to be the scenario anyways, because viruses, as we know, are dependent upon host cells. And I discussed this in my team skeptic debate. And that means that the cell's genome would have then had to predate the viruses themselves, because viruses, as we know, are not capable of surviving without a host cell. And you can read a lot of technical literature coming from the creationist side on this, especially due to genetic entropy, genome degradation. Some of these ERVs could also be the rogue products of the genomes of their hosts. And the parts of the genomes that say we point to that look at like ERVs are then seen as designed to do what they do, and that's where we find the function. But like you said, some of them are damaged due to genetic entropy. Of course, we would never say the entire genome is functional, but definitely a lot more than the evolutionists would say. But even just going back to basic science when it comes to viruses, evolutionists have a totally different understanding of what viruses are. They think viruses are bad when in fact viruses are extremely beneficial. They'll say, viruses beneficial, created by God? Don't viruses cause disease? They'll say. And this is the brainwashing, okay? Because the answer is no. What we know is viruses are incredibly functional to our bodies, just like bacteria are. But it actually turns out we have more viruses than we actually have bacteria. Okay. The question is, what are these viruses doing? Well, the evidence suggests that a lot of them are controlling the number of bacteria in, say, our gut, and also the number of species of bacteria. It's all about regulation. I discussed this in my debate with Drysdale, and he was fascinated and caught off guard, but this is just basic science. The viruses are actually regulating the number of bacteria. And we also know bacteria are incredibly beneficial as well. But due to the fall, due to degeneration, of course, these things like bacteria, viruses, they can become bad. They can become damaging. But the vast majority of viruses are not damaging. They're beneficial. And we know, for example, if we look at an analogy, a hammer can be used for good, for building things, for example, but it can also be used for bad. You can use it on somebody, which wouldn't be so good. And that's what we're seeing with viruses and bacteria. So without viruses, there wouldn't be any regulation. There would be an imbalance in the bodies of living systems. There would be an imbalance in the ecosystem. So no, viruses are good. Most of them are good. Some of them are bad, of course. You can look at the viruses that have jumped from one species to another and burn hot and fast, of course. But, that I just wanted to cover the fact that the evidence does suggest, if you want to touch on that, Matt, the evidence suggests that these are all built-in DNA elements. And here's the thing. I'll say, are these created units of DNA function? And then they automatically just dismiss. They think we're just dismissing how a lot of these DNA elements do resemble viruses or even pseudogenes. They've named these elements in our body, genetic mistakes like the pseudogenes. No, we know they're highly functional. Or ERVs are just useless ancient remnants of viral infections. No, they are functional DNA units and DNA elements. We don't always have to adopt the evolutionist terminology because we know they're wrong over and over again, especially when it comes to function and junk DNA. Go ahead, Matt.
Yeah, I just thought I would touch on something since you kind of mentioned viruses and bacteria, and I thought it would be important to bring up because a lot of people actually blame God for a lot of these things causing problems in humanity. And an analogy that I want to bring up is I want you to picture what bacteria's job in nature is, the vast majority anyway, and that would be to do their job, what they were created to do. So picture an elephant dying in nature. The first thing that's going to happen is, of course, wildlife is going to eat it. But let's just take the scenario that it died in your living room. Well, there's enough bacteria on the elephant, and in the atmosphere, basically, that on the ground, that it would actually eat that entire elephant away given enough time. So that's his job. But what happens now when we say take crude oil and we create multiple different elements, like let's say plastic, which is synthetic. So now we have like a plastic bottle, which never existed in nature, and we throw it in the environment. Well, now that bacteria comes along and tries to do its job, and it can't do it. It can't eat the plastic. So what it does is it mutates. Now we get things like flesh-eating bacteria.
Right.
That's more dangerous because it mutated into a harmful thing trying to actually do its job. It's not God's fault. It's just what the thing was trying to do got a mutation, made it worse, and now all of a sudden we have people blaming God for flesh-eating bacteria. So
It's a great point. It's an amazing point, and what's sad is most of the brainwashed evolutionists have not heard this before. Bacteria, we know, and you and I are like-minded. Bacteria are highly beneficial to our immune system, the health of our bodies. I often take probiotics. Good bacteria, especially if you're going to take antibiotics that's killing all your good and bad bacteria. You better take some probiotics after that fact and replenish that bacteria. Bacteria is good. And viruses, like I said, they don't generally cause disease, even though evolutionists seem to think that way the second they hear virus. They kind of run scared. And the thing is, that's why the evidence does seem to suggest that when it comes to the viruses that oftentimes look like ancient viral infections, okay? We now know they're tightly regulated into our genome and also highly, highly functional. Therefore, the evidence is on our side that suggests they were created this way on purpose by the designer. There's no way around it. And we got to be plain. We got to be clear here. Yes, there are some viruses that are bad. We're not denying that fact. For example, when they jump from a different species, the new species, as we know, oftentimes does not have the ability to regulate the virus properly. And you can see viruses like Ebola, coronaviruses, for example. So these would have been viruses that jumped from its originally created arena and is now in a brand new environment. And that new environment oftentimes is us, humans, and our immune systems don't know how to deal with it, So when it comes to viruses, when it comes to these DNA elements, we can have internal and external escapees. Okay, an internal escapee can simply be due to mutation, like you said, genetic entropy. It's all evidence for the fall. So these evolutionists, when they hear the word virus, think good. When you hear the word disease, think bad. But most viruses don't cause disease. So I just wanted to point that out. And what's funny is I pointed this out to Erica, and she's kind of becoming like the snake was right in the emo now. I definitely like her, but I'm also questioning her objectivity in all this, because then she says, it's the patterns. We share more of these DNA elements with the chimpanzees than we do with other less similar animals or creatures to humans. Of course, we're going to share the most of these created DNA units with chimpanzees. We share the most with the chimpanzees in terms of anatomy, morphology, genetics, physiology. This is just basic common sense. And Matt, you and I were talking about this earlier when it comes to her being so adamant, to call humans apes, just like, humans, we know we're mammals. We're vertebrates, we're eukaryotes. This, just because a pine tree and a whale and a human are eukaryotes does not automatically mean that they are related. We classify things all the time. Taxonomy is a classification system made by man. And yeah, humans are mammals, and we also share the most in terms of morphology and anatomy and genetics and physiology with the apes, like the chimpanzee, bonobo, of course, orangutan, and gorilla. But we discussed this, Matt, okay? And we've asked this question. I've asked this question in the comment section 4 or 5 times, no answer. Do they expect God to use different building blocks, different building materials, something different than DNA, RNA, proteins, carbohydrates, whatever, you name it, no blood, bones, muscles, to have created humans? Do they just expect humans are created totally different, totally equally distant from every other form of life? Or I like what you said, Matt. Do they seriously think humans were created by God as like an empty vessel, no built-in information? These are questions that they really need to ask themselves. You know what I'm saying, Matt?
Oh yeah, of course, because you're going to match something. Everything does in life. It's just the way it is. If we're going to invent a mode of transportation, it doesn't matter if it's a rollerblade or a skateboard or a bicycle, they are going to have wheels, because that's the type of transportation that we make that's convenient for us to do it. It's one of the faster ways that we've done it, remember? So of course we're going to say, well, there's similarities, but they're not identical, but they share them. It doesn't mean much beyond that. It's kind of like, well, if we're going to go out and we're going to create something new, guess what? We're probably going to have an invention like a Segway or something else that's going to have wheels until we come along with some maybe super advanced technology where we can hovercraft or something. Who knows? But I'm just saying that the general thing, we don't create something that's a mode of transportation that's so astronomically different.
Right.
That
right.
we copy off of what was already there. It's pre-existing. We just keep going off of it, and we further and we better. You've seen the original pictures where a caveman inventing a wheel, right, from something square, and then keep improving on that because it's what we use. It's the same thing.
Well, it's just funny because it's common sense that when you're built with DNA, RNA, proteins, you're built with similar building blocks, similar building materials. Of course, then as humans, okay, when it comes to genetics, anatomy, morphology, and physiology, we are going to be more similar to some creatures like the chimpanzees, like the other types of apes, of course, gorillas, bonobos, than some other creatures like fish or bacteria. This is just common sense. Now, evolutionists will say, well, we're less similar to the fish than we are with the chimpanzee because we share a more distant common ancestor with the fish. Okay, fine. This is why nested hierarchical patterns and homology and mosaics, these are all agnostic to the debate. That's why we like to focus on that, which is differentiating. But it's just ridiculous that she's so bent on wanting to call humans apes when it doesn't prove that humans are actually related to the apes. It would be like saying, yeah, humans are mammals. We have the characteristics that classify us as mammals. That doesn't mean all mammals are therefore related. It makes no sense. Now, here's what's funny. The second you can agree that, especially with theistic evolutionists, this is what's funny. Dr. Joshua Swamidass, okay, he's pretty militant against the young earth creationists, but he's actually come out and at least admitted that it makes no sense that God would have created Adam and Eve homogeneous. It made no sense to create Adam and Eve in the biblical kinds with no pre-existing functional DNA differences, because it makes no sense from a creation perspective for a creator to design that way. That's not optimal design. Adam and Eve were to create clones. Makes no sense. He's actually come out against people like Vanima Collins. So people like Erica, Dr. Dan, whoever wants to use that argument that created heterozygosity is post hoc. No, it's the best explanation, the most reasonable explanation. That means when you have that front-loaded genetic diversity, okay, in Adam, Eve, and the created kinds, guess what? That necessitates hierarchical patterns in anatomy, morphology, physiology, and genetics. If a scientist were to show up to the creation event 1 hour after God created Adam, Eve, and the biblical kinds, but they didn't know they were just created, and they started doing assessments, they started doing DNA analyses, DNA sequencing, guess what? They're going to conclude nested hierarchical patterns, and they're going to conclude some type of descent with modification. But guess what? They were just created that way an hour prior. This naturally flows, but it also leads to testable predictions. Here's one thing, and I challenge anybody who wants to fight the starting position of created heterozygosity by giving a better alternative, which would be origin of life naturalistically, abiogenesis, which is an absolute fraud. It's been destroyed. I challenge them to watch the interview we had yesterday with Dr. Hayes, where Tony, myself, and Dr. Hayes destroyed abiogenesis. Give us a better plausible alternative. They're not going to, because they're not going to be able to answer the question, okay? Right now, and I'm going to go over a few points that we discussed yesterday just to kind of put this to bed. And if Erica's watching, or if she does watch, answer these questions. If you are going to say that created heterozygosity is not a plausible starting point. We know, and we discussed this yesterday. So in a nutshell, the most common scenario right now for the origin of life is called the RNA world, okay? And what they do is they propose, I should say, imagine, evolutionists hope, dream, and imagine, right? That non-living chemicals arrange themselves into the first cell, and that cell is the ancestor to all living organisms today. Well, they imagine you have this ocean or lake full of RNA molecules that somehow learn to reproduce themselves, slowly evolve, morph, change, whatever, into that first reproducing organism. This really is the final straw in origin of life research. The fact is, RNA is more unstable than DNA. Okay, and we discussed this yesterday. The fact that in our cells, there are about 1 million DNA breaks every single day. And guess what? Most of these are fixed by the amazing, intricate repair systems designed and built in the cell. Okay, but if RNA is less stable than DNA, as we know, and DNA requires many amazingly designed DNA repair systems in order to survive over time, how in the world, okay, and I'm asking this to Gutsick Gibbon, Dr. Dane, any skeptic that we have, how on earth can an RNA world then be plausible? The building blocks of RNA are threefold, okay? You have a sugar, a base, and a phosphate. Naturally, these things don't bind to form nucleotide bases needed for RNA. So right from the start, what do you have? You have a major issue trying to even get the building blocks for RNA. The building blocks themselves are incredibly unstable. Another thing we discussed yesterday, enzymes are needed as a catalyst in the cell. Yes, RNA can do this in a very, very limited extent, okay? But the reality is that in cells, in living cells, protein-based enzymes do most of the work. How did life, very precise question, I want empirical evidence. I want a scientific explanation, not storytelling, not imagination. How did life change from RNA enzymes, from which there are only limited support for, to this amazing protein enzymes that we have? When did this occur? We have real chicken and egg problems because information in the DNA requires enzymes to read it. But then those instructions to build the enzymes are, guess what? They're on the DNA, which cannot be read without the enzyme. So which came first, the enzymes or the DNA? But guess what? The metabolism first hypothesis is dead. It's been dead for years. And we could go on and on and on. What's funny is we discussed this yesterday too. So this is just everything in a nutshell, but I want to put it to bed. Life isn't even just based on biochemicals. You can have all the biochemicals you want and you're not going to have life. I can give these skeptics, okay, I'm going to give them the amino acids, I'm going to give them the proteins, the RNA, the DNA. For one, okay, they're never going to assemble themselves together. Show us how that's possible. But even if they did have that, we still have not introduced information into these molecules, okay? So now you've got the information problem. Problem after problem after problem, but they want to scoff at front-loaded genetic diversity. It's the more plausible explanation. It leads to testable predictions. Even outspoken speakers like Dr. Joshua Swamidass states that, and I this comes to mind because Erica just left another ridiculous comment saying, you know, I've seen you discuss with Dr. Joshua Swamidass, and it's true. And what's funny about Joshua Swamidass, here's a contradiction if I ever heard one. So he said, you know, he's come out against people who use that argument that God would not have front-loaded Adam and Eve and the created kinds with pre-existing genetic diversity. Of course he would have, okay? So that's a good starting point. Then he tries to say, uses this argument, okay? The most ridiculous argument. Supposedly it's the fatal blow. It's a joke. It's a contradiction. You ready? So he says the TMR4A, okay, it's a problem. It has to do with population genetics. He says this destroys Adam and Eve, okay? All TMR4A is, guys, to the audience, it's a sophisticated version of treating all human alleles as the product of mutation and then tracing them back in time to a common ancestor. Okay, if you didn't see the contradiction, he is now assuming all DNA differences are the result of mutation over time. Okay, these alleles, for example, variants of a gene, of course, flavors of a gene, I should say. So he's harping on people that misrepresent him, okay, and try and say that he's against the created heterozygosity hypothesis. Okay, fine, that's good. He's finally come out and he's admitted to that, which makes sense from a scientific and theological position. Then he uses an argument, Matt, okay, as an attempt to rebut Adam and Eve in the biblical creation model by not assuming the created heterozygosity. It's a circular argument. So he assumes away the premise of created heterozygosity, that he has come out and admitted that this would be the case if God were to create living organisms, in order to refute this. So I don't know if Swamidass has actually realized it yet, but this is a straw man, despite, and this peaceful science discourse is hilarious. You should read through the section where I debated all these guys, including Swamidass. The most pathetic arguments. This is supposedly their best one. He repeats it over and over again, and it's a straw man. I don't know if he realizes it. That was long-winded, Matt, but what do you think, brother?
Well, what I don't understand is you said earlier that Erica said that the created heterozygosity is a retrodiction, and she's complaining about that. Like, wait a minute, your entire model is based on retrodictions. Basically, I mean, 95% of the world's scientists, as you guys claim, are evolutionists, and they're not making any future testable predictions. And you're mad that there's a retrodiction in our model. It doesn't even make sense. Like, literally, the very mutation rates that she harps on are retrodictions themselves from the evolutionists. You see, what happened is they came along and they looked inside the human genome, and they found that there were about 24 maximum fixed substitutions in humans. So what did they do? Well, in 1990, at that time, they believed that the bottleneck occurred about 133,000 years ago, not 200. So what they did is they divided how many substitution maximum there were, 24, by the 133,000 years, and they got one substitution should arise every 12,000 years. That would be true if evolution was true. What did they find when they looked in the genome? Did they find that one substitution was reaching fixation every 12,000 years? No. How about 10,000 years? No. How about 5,000? Nope. How about even 100 years? Nope. See what the problem is? They're in deep trouble. So that's a retrodiction that they made based on their model of evolution assumption being true, and it was completely wrong. They can't even get a mutation rate, even by slowing it down as much as they possibly can, even close to those numbers. So that's what you get with evolution.
Well, that's why Erica has really got it cut out for herself. And because she brought up Joshua Swamidass, why don't you bring that up to him based on his population genetics argument that is actually based off a straw man that assumes away created heterozygosity, which he was vocal in saying that God obviously would have created Adam and Eve with some genetic diversity. So that's pretty sad. She's got a lot of questions. The critics do. I mean, Dr. Dan has failed, but she's got a lot of questions to answer. In our last couple of videos on molecular clocks, for example, molecular clocks and genetic data, at the end of the day, they've officially demolished deep time evolution and universal common ancestry. That's why they fight it so significantly. And then I want to see these answers to the origin of life, these questions that I brought up. Answer the chirality problem as well. I won't even waste any more time on that, but answer those questions. Let's see what these pseudoscience pushers have. Now, let's get into the Y chromosome. Okay. First off, let's talk about a few arguments that we've seen coming from these guys on Dr. Nathaniel Jensen's paper. What are a couple that come to mind, brother?
Well, I guess the first one would be that they blame Jensen for using the regional filter, but Cameron did not in their analysis. So they say, see, he's cherry picking his data, and therefore he shouldn't even be admitting or using Cameron's study if he's not even going to use what he did. So they don't even understand what they're talking about when they say that. I don't know where they got this, because the regional filter is the one that everyone else in the field uses, and the one in which he also used, Cameron I'm talking about. So when they said, oh, Jensen used it and Cameron didn't, he did use it. He just didn't like the results that it got. So he mentions the other ones. So it shows the critics don't actually understand anything about what they're talking about when it comes to that. And here's the thing, Cameron admitted that the results that he got in the regional filter didn't match. So he invented more filters because of that. What does that tell you?
Right. Yeah. It's this is the best they got, and it's sad. And guess what? Never any testable predictions that are coming from them. Never any real comment on, like, for example, Erica was saying today, I might as well just screenshot this real quick, or let me see here. She was saying, "Oh, why did Jensen only say that there are 2 papers?" Did you want to comment on that as I was screen sharing this real quick? I'll show a slide on that. 2 papers, she said. Jensen ignored the rest. What do you think about that, Matt?
Oh, he ignored the rest. Yeah. Well, it doesn't exclude the high or low coverages like they're saying. That's factually incorrect. The math that Cameron used in the paper, it's very clear. So, it's also the filter that the leading experts use. So I don't know why they're complaining that Jensen used it when that is what the major people use. So again, it's just complaining when they don't know really what they're complaining about. Another problem they say is when investigating the Y chromosome, Jensen ignores the remapping filter, which
yes,
yeah, that's another one, right? That's
keep going, debunk that, brother.
Sure. Well, this again, it comes down to the critics not really thinking critically, because the remapping filter is what was invented by the evolutionists to solve the problem of the mutation rate being so high in the Y chromosome, right? So it is defined by the agreement of evolution. It's a circular argument, and no one else in the field uses it. So I don't understand their problem, really. They're not thinking critically with that one. Not at all. I mean, if they looked at it and they saw how biased it was, and they looked without the goggles of evolution, they would actually see they're being biased. But we both know that that's not true. How come Erica can't understand our model? It's because she's invested. How much money has she thrown into college and her future career? It's obvious that she can never admit that, oh, I just wasted how many years of my life studying this nonsense and given money to just wasted. Her whole future is determined by what she wants to do with the field of evolution. You think she's ever going to acknowledge that it's a lie? Of course not. That would be ridiculous. You'd literally just admit that you just threw money down the drain. But anyway, let's get back to the critics, right?
Well, and because she's so militant and she's been a little bit more aggressive in the comment section, and I still respect her. I like her. I'd have a coffee with her, but I'm just going to be straight up honest here. There's a lot of damage control going on, okay? Because she came in here, saying she's been at this forever. Young Earth creation has no arguments. Going right back to my first debate, her expertise is human evolution. My entire opening and my entire premise had to do with the post-Babel dispersal of the people groups that experienced isolation, inbreeding, and rapid genetic degeneration. In that debate, I also talked about how habilis and sediba, that there's strong evidence that they are the result of artificial species. Habilis is a joke, nothing but fragments. It's actually kind of funny. I brought up the Y chromosome dissimilarity. I had some questions for her on the molecular clocks. You can see she just completely dodged it, avoided it. Same with the chromosome 2 fusion. And like I said, great debates with her. They've been respectful, cordial. Because she's more militant now, I feel like I can say this. And then that's her, but she had no answers to all these things, no rebuttals. She didn't even comment on the hypothesis that suggests that these hominins like Neanderthalensis, Erectus, Naledi, Floresiensis, Luzonensis, these are all the result of rapid accelerated genetic degeneration, explains all the so-called primitive features. Then 6 months later, she starts coming out with these videos. What about 6 months ago? Y chromosome, same thing. Next debate, orphan genes, endogenous retroviruses, molecular clocks. Her response to genetic entropy was a written rebuttal, but apparently she's been at this for years and years. Nothing against her, but that's the thing, Matt. Don't you find that these people who've been at it for years and years, but then they have no answer? It's like I brought up the out of Africa problems. Okay, that show that the out of Africa scenario is not even remotely feasible. I mean, the genetic damage associated with that hypothetical population bottleneck of 2 to 10,000. She literally said, "I've never heard of this before. This is new to me." Oh, that's because these critics don't read the creationist literature, but then they want to say they're experts on it. Matt, what are your thoughts on that, brother?
Yeah, of course. They say, oh, we're ex-Young Earth creationists. We know all the data. So, I mean, that's why we're ex-Young Earth creation, because we realized it was a lie. It's like, oh, really? Tell us about our model. How does it work? Well, I don't know. What are you talking
about? Exactly. And that's the thing. They're overly confident. They've debunked Young Earth creation. Then you bring up the questions and, it's deer in a headlight. You can see I'm screen sharing. So, Jensen apparently only looked at one study. You can even see in his presentations, he's at least mentioning 4. In his paper, I reread it today. I've got it in front of me. He points out the fact that there's been a time. Here he says 2 early studies based on low-quality DNA, 2 subsequent studies based on high-quality DNA. There's at least 4. If you look at his paper here, because she's trying to say he's ignoring other papers too, right? In his actual paper, now here's the thing, her word-for-word rebuttal was almost from Evograd in the Peaceful Discourse webpage that I'm in, and I recognized it right away. Remember I sent it to you? I said, this is exactly where she got it. So she's going to have to do a little bit of a better job. But he points out that there are other studies that have derived a mutation rate. This is right in the paper, via the historically circular evolutionary geology-based molecular clock method, or by extrapolating the autosomal mutation rate onto the Y chromosome. And the one thing that they always ignore is the testable predictions. And we're going to go over that here in a second, but I just want to point out that directly in the paper, he discusses that. I'm going to pull it up in the paper right here so we can get a direct answer. So right here he says, however, since our results imply that the entire tree was only a few thousand years old, our results predicted that known recent, within the last few thousand years, changes in population size would be stamped throughout the tree in a manner consistent with the recent origin of the tree. In other words, the deeper roots of the Y chromosome tree should record not changes in population size from 200,000 years ago, but changes in population size from the recent past. And then you can read all about and then in another paper, he goes over the fulfilled predictions. So if there's so many glaring errors, but it's all from misrepresentations, why are they working so well, Matt? Why is the active research program working so well? So what I'll do here, for the audience sake, now you can read his papers on here, read his technical papers on here, but I just want to give a brief breakdown of what we're talking about. Okay, so when it comes to the Y chromosome, what we do is we can take father and son, and then we can count how many mistakes occur each generation. Okay, we know mistakes happen every single generation, every few generations. Well, we want to get dad's Y chromosome, son's Y chromosomes, grandfather's Y chromosomes. We want an accurate measure of the rate of mistakes. Okay, they've done studies. They did an initial study, for example, in 2009 and another in 2015 that Jensen specifically looks at. Those were based on low quality data and low quality DNA. Okay, the results that were concluded from these papers and the rate of mutations that were empirically discovered were slow. Okay, they're very slow. But when you look at the 2 additional studies that have been done, okay, one that meet the requirements of, because here's the thing, when it comes to the evolutionary model, you can't assume evolution to disprove evolution, for example. So in the study in 2015, correct me if I'm wrong, man, and there's another in 2017 based on high quality DNA. What they discovered, and you'd have to look in the supplemental material oftentimes for this, is that the number of father to son Y chromosome differences were approximately 10-fold higher. This was unexpected on the authors of the paper too. They admit, Matt, we always laugh about this. What they admit? They admitted that the empirical findings prompted them to what? Explore additional filters. Isn't this what they always do, Matt, in the mtDNA? We're not going to touch on the mtDNA here, but we did in great detail in the last video. Isn't that what they always do? Explore additional filters, calibrate the data, come up with new rescue devices. In other words, for the audience, what they did was they took their high quality DNA results and filtered stuff out that doesn't fit their evolutionary timescale. Isn't that right, Matt?
Yeah, you're right. It's pretty ironic, like they've always had these little sequences. So with people listening, the mitochondria is much smaller. It's about 15,569 base pairs. And the Y chromosomes, 58 million, right? So obviously sequencing that is extremely hard, but over the years, they get more bits and fragments and bits and fragments. So finally, a couple studies came out where they actually took all these and they bound them together. So they have huge fragments that they can now look at. And like he said, they looked at it and they found that the mutation rate's high. And it was so high that the what they calculated and that they got didn't match. The most recent common ancestors for males was 4,500 years ago. So what they do? Boom, new filter. That's not good.
Right, exactly. Well, see, it's frustrating because the Y chromosome has been typically what, low coverage sequencing. That's frustrating because the error rate in the data is high enough to the point that it can't be trustworthy on every single letter. So, for example, is this a mutation rate from one person to the next, or is this an error rate in the data? So, the high sequencing data that has been looked at has revealed that there's only been 4,500 years worth of mutation accumulation. Erica's arguments are debunked directly in the paper that she says she read. I suspect she just ran to Evograd or whoever to get an explanation. But in a nutshell, you can watch, what is it, Matt? It's almost a 30-part series on the history of civilization. Stamps found in the Y chromosome.
Oh yeah, it's huge too. They're hours and hours
Each video. Why is it working so well? This didn't all have to be true. So in a nutshell, watch those videos, watch his paper where he goes into incredible detail. Once again, for the audience sake, I'll break this down as simple as possible. Okay. For the Y chromosome, the clock ticks every single generation. So in theory, okay, we should be able to walk back through every single of these 200 or less generations to the beginning, according to our model. We should be able to, if this Y chromosome data is true, okay? This is why they just nitpick on little things, but they never address the key issue. We should be able to see each generation of history in this Y chromosome family tree. This is fascinating. This won't be addressed as always, I guarantee it. The branches of a family tree reflect population sizes. Now, if we were to make a Y chromosome family tree of the globe, for example, the number of branches at any particular point in that family tree should then what? Well, it should reflect the global population size. As we talked about earlier, we know DNA mistakes occur. And in the Y chromosome, mistakes happen every generation, means we can reconstruct a global family tree of humanity because DNA is a record of the global family tree. And this is what's a shame is in our third debate, me and Erica, I brought this up. And apparently she just finally went and read it, yesterday. So all of this time, just dismissing the evidence, dismissing the confirmed prediction. Same thing in the mtDNA, genetic stamps and signatures on the history of civilization. And we go into this in incredible detail in the other videos too. So as I was saying, DNA is a record of the global family tree. And what Jensen goes over in this series and in these papers is that global family trees record changes in global population sizes. Therefore, the global family tree is then a record of the global change in population size, okay? Now what we do, stick with me, is we can test if the world family tree is a record of long periods of time, aka deep time evolution, okay, which they don't want to make predictions of their own. Because remember, if we really do go back to, say, a last Y chromosome ancestor of 200,000 years, even though their dates are all over the place, or an Eve of same thing, 150, 200,000 years, then this last 6,000 years of human history it's like a needle. It's just the tip of it. It would be nothing but a scrambled mess. Okay, for the most part, you're not going to be able to detect signatures. But if it really does just go back 6,000 years, both Y chromosome, mitochondrial DNA, we should be able to detect the history of civilization, okay? Now, that's why we would say it's a shorter period of time. This 4,500 year history, according to the Y chromosome, it would be Noah, of course, if it's true, should be stamped all throughout the Y chromosome in the form of, as I've been saying, genetic signatures. The global Y chromosome tree is the key to human history. And guess what? Guess what's being detected? You got it. All kinds of genetic signatures in relation to historical events in the Y chromosome, which confirms predictions made by Dr. Jensen. So is Erica or the other critics, are they just going to dodge it yet again and just copy the blogs, or Herman Mays, which has been addressed, Dr. Frello, he's been addressed. Most of these people just want to kind of throw out arguments, even though they don't know what they're talking about. They don't want to be held accountable. They don't want to submit their objections to the AIG journal, where Jensen will then respond. He's responded to Frello. Frello's the only one who's taken up that offer. So are they going to address these key issues, this key data? Once again, if there's so many glaring errors and omissions, Why are they working so well? Isn't that the key issue, Matt?
Exactly. And here's the problem. They like to nitpick. They like to complain. Why did he just use Carmen's study? Well, guess what? Why do the critics ignore Moretti at all? Remember, his results with the Y chromosome also confirmed Carmen's mutation rate. So we have two separate studies using high quality Y chromosome that both converge and state the exact same thing. And they both hide the data from the public. What does that tell you? What does that tell you about evolution?
Tells me it's a fraud. It's not predictable. It's no longer falsifiable since they force fit. Matt, just look at their published dates. Okay. Once again, to the critics, Gutsick Gibbon, whoever. Do you not know that the published dates are extremely variable? Sometimes they range from 50,000, some of them to 300,000 years ago. And Matt, what do they base this off of, right? A lot of calibrating, really, really odd, sketchy archaeological data. They rely on assumption-based methods to what? Calibrate these molecular clocks. That goes for the mtDNA and the Y chromosome. So just looking at that huge range of error on the Y chromosome atom, okay, should make you wonder, why aren't they making these testable predictions? Because they have no consistency. Okay, and here's the problem. These assumptions, convoluted assumptions, oftentimes lead to these discrepancies. Now, here's the thing. The data actually, if you want to look at the bigger picture, okay, just like the mtDNA we covered the other day, we're not going to rehash that, but there's only about 300 SNPs that separate modern man, the average modern man, I should say, more or less, right, from the Adam sequence. So all we have to do is assume a Y chromosome mutation rate of about 1 per generation, right? And the most empirical use of the molecular clock would put Adam, well, according to our model, it would be Y chromosome Noah, exactly where we would expect 4,500 years ago. You can assume a generation of 25 years. But here's the thing. Here's what's funny is the dates. Sometimes we were talking about this sometimes because you have to consider population histories, population sizes, generation times, lots of different things, right? Because we know small populations, especially for the mtDNA, mutations can be fixated rapidly. So even if you get a date between 5,000 and 10,000, it's right in line with young Earth creation expectations. How do you get those dates to the deep time evolutionary requirements, Matt? How do you get to those dates? You can't. That's why they won't make the predictions. Here's what's funny too. Here's another thing. Okay, when it comes to the chimpanzee, the Y chromosome for the chimpanzee. Okay, when they first discovered this, and Erica's been totally off on this. I'm going to destroy her on this right now. Instead of being nearly identical to the human Y chromosome. Okay, once again, inconsistent, unexpected, not predicted. Radically different is what they discovered. Not only that, the chimp Y chromosome is only half as long as the human Y chromosome, meaning there's actually less than 50% overall similarity. Okay, the remainder of the chimp Y is only 70% similar to the corresponding part of the human Y chromosome. So total similarity is actually far less than the 70%. It's probably between 40 and 50. From the deep time evolutionary perspective, ape to man evolution, to get this much divergence in, say, 6 million years, their numbers are a little bit different here and there, would require an incredibly high mutation rate for the Y chromosome. And the authors in that paper, you can read it, they claim that the chimp human difference, Matt, do you remember this, is more like they would have assumed or expected when comparing the genomes of humans and what? I believe it was chickens or birds.
Chickens.
So, and according to their story, humans and birds would have, the common ancestor, hypothetical common ancestor, would have been roughly 300 million years. So this is how shocked they were. There's no way. Now, here's the thing. A lot of evolutionists, they'll agree Y chromosome mutates fast. They needed to mutate even faster to make up for the incredible dissimilarity, okay? Even if you want to go with a conservative number like 70%. So now those 300 mutations that we are removed from the Y chromosome sequence, okay, are easy to explain. Because if the Y chromosome mutates extremely rapidly, like they say they do, just to explain that incredible dissimilarity or changes rapidly, okay, then it makes more sense because we know that there's incredibly low variation in the Y chromosome. There's no highly divergent or ancient Y chromosomes found anywhere. Erica sent me a study on ancient DNA, which is exactly not what I was asking for. And African Y chromosomes, just because they may appear to be the most ancient out of the human population, overall, there's extremely low variation. There's no highly ancient looking Y chromosomes. If this case were to be true, if the human Y chromosome has been changing rapidly for however millions of years, okay, since the split, there are things we should find. There's predictions we can make, and we don't find those things. So it's erroneous. It's wrong. So it's even easier to explain those 300. There's not a lot of mutations. When you look at a phylogenetic tree on the mtDNA Y chromosome, there's not a lot of mutations. Incredibly easy to explain in a biblical time frame, and coming from two distant common ancestors, according to our model, in the recent past. But according to the evolution model, problem after problem. They're going to have to invoke tons of purifying selection in order to lower the overall substitution rate, of course. They don't like the raw data is the problem. So it's just so funny that, yeah, okay, if it does mutate fast, that's exactly what Jensen's finding too, based on the high quality DNA sequencing data, then the fact that we're all incredibly similar, we have low genetic diversity in the mtDNA Y chromosome overall in our genetics, then that means it has only been mutating fast for what, Matt? Just 4,500 to 6,500 years. That's the only explanation. They agree it mutates fast. They have to say it changes fast, given the dissimilarity with chimpanzees. What are your thoughts, brother?
Well, yeah, they're stuck between a rock and a hard place because of that data. But like you were saying earlier, too, they're because they rely on the phylogenetic method, which is the assumption that the geologic column is correct. They basically create these mutation rate studies based on the inference and assumption that these geological record layers are true, and that the man split from a chimp ancestor about 6.5 million years ago. So they have assumptions with that being true. They have assumptions with original populations. That's why their error range is so big. Like, you know, the mutation rate, the observable mutation rates are errors of, you know, thousands of years. But then again, when you get into these phylogenetic studies, like look at the 1991 Vigilant et al. Study. He was 166,000 plus or minus 55,000 years. Then in 1992, Stone King and Sherry came along and they obtained 133,000 to 137,000 years. Then in 1995, Horel and Hayasaka came along and they got 143,000 plus or minus 18,000. Then in 2004, Lowe came along and got 72,000 to 108,000. Then in 2012, Skelly and Durbin came along and got 250,000 to 300,000. Then in 2013, they got 157,000 plus or minus 40,000. Then in 2019, they got 286,000 plus or minus 32. Look at that. That's an error range of 72,000 to 360,000. That's insane. Look at those. You know what I mean? That's just
amazing.
Throughout the years, it's varied so much. They just keep changing it around. Nothing matches. It's like there's bottlenecks just everywhere. They just throw them around. There's no consistency. Unbelievable.
It's unbelievable, and great points you make. Okay, so in Erica's video, one of her videos, and you know, we've addressed everything thoroughly. Now, here's the thing. Actually, Dessel Drace, where did the show go down? I missed her comments. Go to it's titled Evolutionists Still Don't Understand Orphan Genes, my good man. I pinned her comment. There's probably like, what, 40, maybe 50 comments now. So go read through it. There's some good stuff. We've responded to everything, and now we're just kind of going into more detail. But in one of her videos, Matt, what did she say? We laughed when we saw it. She said, we just destroyed the Y chromosome part, dissimilarity. But what did she say about chimpanzee and human Y chromosome? She said that, I think the paper she had was a 2015 one. I'm not sure. I'm going to put it in the, I've got a ton of papers. I probably read through like 10 papers today on this issue. And in this one, which is a 2020 paper, she said in the video, go watch it. I'll link it too. Actually, everything's linked in the description. She said humans and chimpanzees, even though they are so dissimilar, is still the closest out of all the great apes. Do you remember hearing her say that?
Yeah, which was great because it got you into finding another study that was done this year.
Here it is. I'm screen sharing. Can you see it, brother?
Yeah, I see it.
So what does it say? 2020, read through. This is great. Puzzlingly, in terms of shared genes and overall architecture, let's keep reading. The human Y is more similar to what? The gorilla Y than to the chimpanzee Y, even though human and chimpanzee have a more recent common ancestor. Y chromosomes from additional great ape species should be sequenced to understand whether interspecific variability in gene content and architecture is characteristic of all great ape Ys, and not just of the 3 Ys previously assembled. So really, they only have high sequencing data on the human and chimpanzee Y chromosome, and that's where we find Brother Nicholas made a point that based on the numbers, it would be like 35%. Are you kidding me, Erica? Come up with something better than this. And this is a 2020 study. You got to be up to date on these things. Now, here's the thing. The bonobo, I've been asking, okay, so are you trying to say, because I would predict, Matt, would you predict? Because we would say, kind-wise, you're looking at the human kind, which we've already separated as its own unique kind based on a number of things, molecular clocks especially. Then you've got the bonobo and chimpanzee who've descended from a common biblical ancestor kind. Then you would have the gorilla, and you'd have an orangutan, and then you'd have an Australopithecine kind. You know, you're looking maybe 5 or 6 different kinds of apes. And a human kind, of course. She wants to play her taxonomy games, but it ain't going to cut it. So here's the thing. We would predict that the chimpanzee and bonobo would be more similar in the Y chromosome than the human to the chimp or the bonobo to the chimp. And there's no exact number because there hasn't been full sequencing of the bonobo. And I've been asking, where's the number? You can Google what is the % similarity between humans and chimpanzees. And because we're looking at high quality sequencing, we've got the numbers. So she failed miserably on this argument. I've been asking this Y chromosome question for months and months. I'll ask it to Dr. Garrett. You know, people are baffled by it because it really does show us strong evidence in combination, though. We look at the orphan genes, the DNA function, of course. Incomplete lineage sorting is an issue. But also an argument she was trying to utilize is higher genetic diversity. Remember, Matt? We know species within chimpanzees, okay? Different chimpanzee species or subspecies have more diversity within themselves than humans and Neanderthals do. Okay, do I need to repeat that? And they want to say Neanderthals are a separate species to Homo sapiens. Are you know, and this is why we always say that the data is consistent with the fact that God created 2 human beings, Adam and Eve. They would have only needed roughly 10 million variants. Explains everything from there. Mutations post-Babel, post-flood, of course, can easily explain some of the highly variable positions. I responded to Dr. Dan on that. I've had no good response because there is none. They're not up to date on the literature. So everything we find is exactly expected. Yeah, there is higher diversity in the pan genus, being chimpanzee, bonobos. There'd be higher diversity in the gorilla. So this is just hilarious. So some quotes from all these 10 papers that I've unless you have any thoughts, brother, but I've got a lot of quotes from some of these papers that I found today. So here's one. Studies of Y chromosome diversity in other great apes are relatively undeveloped compared to those in humans, just like I said, but have nevertheless provided insights into speciation, dispersal, and mating patterns. And what we find is a lot of rescue devices to explain all these discrepancies, right? This one here, the Y chromosomes of great apes represent a particular puzzle. Their gene content is more similar between human and gorilla than between human and chimpanzee, even though human and chimpanzee share a more recent common ancestor. Here's another quote. Analyzing this data set, we found that the genus Pan, including chimpanzee and bonobo, experienced accelerated substitution rates. You see how they include genus Pan and they're using the same types of explanations? Because they're clearly more similar than they would be to human. I mean, this is just ridiculous. Right here, among great apes, the Y has so far been assembled only in human, chimpanzee, and gorilla. And she wants to make all these conclusions on bonobo. Well, we can make a prediction right now that it's going to be overall more similar to chimpanzee. Here's another quote from, maybe she only read one paper, I don't know, but I went to town today. For instance, the chimpanzee Y is only half the size of the human Y, and the percentage of gene families shared by these two chromosomes, 68%, that split roughly 6 million years ago, is similar to that shared by human and chicken autosomes that split 310 million years ago. Puzzlingly, again, you notice that word again? Let me see again. They go into, here's one. Well, prediction kind of confirmed, but like I said, we need more sequencing, high quality sequencing. But for the bonobo, what they have so far, for instance, it says the bonobo Y assembly had the highest percentage of its sequence aligned to the chimpanzee Y, just like we'd expect, than to any other species, in line with the recent divergence of these two species. Okay, yeah, they might have more genetic diversity, of course, as we know, within those species, exactly what we'd expect with animal kinds, of course. Here's another one. In terms of sequence identity, as expected, the chimpanzee and bonobo Ys were most similar to each other. 99.2% and 98% from multi-species and pairwise alignments, respectively. Like, you can't help but just laugh, eh, Matt?
It's like they're wearing goggles or something.
Oh, it's so funny. Well, I don't want to. I'm beating a dead horse, but just quote after quote after quote. Erica's just going to have to do better than this. She's now relying on Dan's most recent failed attempt at orphan genes. Those papers were hilarious. Nothing but inference, assumption. Once again, asking for, in the lab, a series of mutations that has demonstrated a non-functional region of the genome popping up to be incredibly functional and essential in the genome, like an orphan gene. No, haven't been provided it. Yeah, I've got quotes and notes on the, right here. Among the great apes, all four genera, gorilla, pongo, pan, and homo. Let me see. Only the chimpanzee and humans have fully sequenced Y chromosomes. So, yeah, you can go watch her video where she says that, just gives contradictions to the actual data itself. So now here's the thing. Go read that comment section. I'm shutting it down because I'm tired of answering the same stuff and covering the same misrepresentations. And praise in the house.
What up?
I don't know. I think that was a complete destruction and impromptu too, Matt. Well, I told Matt, I said, you know what? Sick of these comment wars. There's 50 comments. Go look. We just demolished plus some everything. So we're going to go on record here right now. Matt, this has been fun, but we've got a lot of projects. We've got a couple books coming out. We've got some good stuff for everybody on some of your more popular channels, but still what's funny is they don't have as good of arguments though. Like Pelagia, Vice Rhino, Aaron Ra, like they're all pretty science illiterate, right? None of them will debate, but they're bigger channels. They have more influence. So we're going to be destroying their videos. So Erica says she's coming out with another video. But to be honest with you, if it's more of the same, misrepresentations, not addressing anything, asking the same questions, claiming we didn't answer them, even though we've answered them 100 times, we're probably not going to respond for a while. I would just say, hey, listen, because if you look at my description boxes for these, Matt, incredibly detailed. We've got all her videos. We've got our playlist, our videos. They're not going to be convinced. Erica's not going to be convinced. She's invested. Dan's invested. It's for the viewers, the audience. And I say, I'm confident to say it, especially with Dan. Go watch their videos, go watch our videos, and then come to an informed conclusion. You'll see who's dodging. You'll see who's presenting the data. You'll see who's making the predictions. It's pretty obvious. So it's not just going to be an endless war. I'm confident with saying, now, if she brings up something new or something worthwhile, yeah, we'll respond. If it's the same stuff, I'm just going to say, hey, I'll post your video in our description box, but you're leaving it up to the audience at this point. Yeah, that was long-winded, but man, I just thought that was amazing, the Y chromosome. So go ahead, brother. What are your thoughts?
No, man, I mean, you wrapped it up pretty good. Like we've been having, we've been playing ball back and forth, back and forth, back and forth. I mean, obviously we're not getting anywhere. So I guess we just got to move on. I mean, we just went back and forth in the comments section multiple times. I mean, it just gets redundant. I can see what you mean.
And look, Nicholas, amen, brother. He says, good prediction. I agree. There's us doing testable predictions. If we're going to say that humans are their own separate kind, you're looking at chimpanzee and bonobo. I think it's a pretty safe bet to say. And here's the thing. I've asked her numerous times for the exact percent similarity. And I haven't been given one. And that's because I don't think they have an exact number, but I would safely predict it's going to be closer. Well, I've already given quote after quote that shows that they are closer. There's right here in 2020 suggesting that the human and gorilla are closer, which makes sense, different kinds. And Dessel Dre says, but it is an endless war. You're right, bro. You're right, my man. It is an endless war. So it's never going to end. He says we live in a war forever. We love the war. We love the battle. It's a daily battle as apologists. We're always standing for the truth. We're always fighting for the truth, fighting lies, fighting misinformation. So I should correct myself. We just have a lot of different projects now that we definitely want to get some of that done. And then eventually come back to people that we've already, spent 1,000 hours on. Does that make sense, Matt?
Oh, yeah, exactly. The time it takes to make videos, you would be surprised, because when I had all day to make them, I was literally making almost 2 a day. I didn't realize right. Hours a day making videos, but now that I don't have the time, it's actually harder to put them out. So we're going live a little bit more, which I guess the audience really likes. And it's a little easier to just address something immediately when we see it. So I kind of find it fun, but
it's a blast.
It is fun. I mean, people should just leave comments like, what do you want to see as far as a video? Because we're going to start debunking people that we've had in line for a while. But if you have any particular things, just mention it. I'm sure we'll cover it.
Snake.
Snake was right. Yeah, poor guy. He's having trouble passing the 100 subscriber mark. But we've been debating him and giving him attention for 2 years. I think I've had 12 debates with that guy. And what's funny is I've asked, because for Dessel, I've asked Dan oftentimes. Before I gave a Neanderthal response, no joke, I messaged Dan, emailed him, commented. I said, why don't you come onto my channel? And let's be fair. I love people to see both sides and come to their conclusions, okay? I'll give a 15-minute presentation. You give a 15-minute presentation, followed by, I don't know, a 5 or 10-minute just response, and then leave the video up and people can come to their own decision. He didn't want to do it. And so be it, fine. But I don't know what there is to be scared of. But Nicholas here says, it will be far closer between chimp and bonobos. I'm not sure evolutionists would be surprised, but that 35% is damning. It's true, Nicholas, it's damning. Go look at their explanations for why it's the case. People like Gutsick Gibbon, they're never going to admit how they leave a lot out. Okay, and that's the problem. When you read these papers, the authors, their guards are down. They talk about how shocked they were. They talk about how surprised they were. I've got this 2020 paper here still talks about how little they know, how little they have sequenced of most of the apes. They got the human and chimp, and that's how we know how damning it is. So, yeah, we provided a lot here in terms of predictions. She says there's no predictions in DNA function. Well, my question is, give me one prediction Nathaniel Jensen made on DNA function, because I could right now spend 15 minutes going through that. But you know what? The fact that 6 months, this specific critic did not did not actually look at those papers that I brought up in that debate. Kind of says that maybe these people aren't looking for answers, right? But the predictions are there, especially on specific predictions in regards to nested hierarchical patterns, DNA function, and highly conserved mitochondrial DNA proteins. I explained it for 10 minutes to RJ Downard, and he looks so confused in the debate. Like, these guys don't know what's going on.
Yeah, I made predictions as well, remember, on DNA function, because when I looked into the CO1 gene, my determination for determining what function was when these particular genes get a mutation, they affect this particular region of the body, and they're different in different animals. So, for example, the CO1 gene in, let's say, a bat does the same thing that it does in a human. Its main function is respiratory. So, but what happens is when a human gets a mutation rate there, or I'm sorry, a mutation there, or when a bat does, the functions are very different. For example, it affects the brain of a bat, but it affects a human in a different way. So I predicted that obviously the function of these different genes that are inside the mitochondria and in particular the CO1 gene fragment would be different in animals, and I based the function on what would happen with a mutation. So,
right.
Boom, easy, right?
Oh, it's amazing. That's why we have predictions and retrodictions that are flowing from our explanations. Dessel, I want to compliment him. I'm not even sure if he's evolutionist or creationist, but I think he's evolutionist, but he's objective. I like his comments. He watches debates objectively. I think he gave a great review of the Kevin Anderson and Jackson Wheat debate, but he said, yeah, Dan, cutting you off, making rude comments, ending with go do your homework. Well, that's the thing. When it comes to like Dan, the best way to discuss with him, because you can even watch the Sal debate. I mean, he's constantly cutting Sal off, is you need timed rebuttals, 5 minutes, 5 minutes, 5 minutes, 5 minutes. Eric and I actually have never done that. We've had almost like hour discussions each debate because she is good with being patient, not cutting you off. But people like Aaron Ra, Dan, these guys need time rebuttals. They hate when their worldview is getting destroyed.
Oh, wow, yeah.
In front of a live audience. Remember that one video of you on non sequitur? I was showing that to my brother today. We were dying of laughter. Every 2 seconds, Aaron Ra, remember?
Yeah, you can't get a word out. He'll ask a question and then pause you and answer it.
Yeah, exactly. It looks like we've got a lot of super stickers. Paying for my coffee addiction. Doki Doki Bible Club. Like I said, we love the support. Praise! It's your birthday. The big 19. How's it feel? Going out partying or what? Well, with that music, we started with the intro. That's Club Standing for Truth, brother. You're here to party.
Yeah, man, it's sweet. I finally got some hair under my armpits.
Not a boy. No hair on the legs yet, though, eh?
No.
Yeah. It'll grow in. Yeah. Not a boy. Birthday cake or what?
No, just some candles. That's about it.
Some candles.
Fireworks. Yeah.
I thought you were a little bit older than 19, but okay. Dessel says he's gonna go read that thread. Praise, have a good birthday. Matt, where's your paper? Well, we've got it in our newest book coming out on mutation rates, and I think people are going to be pleasantly surprised. We'll have that out.
Yeah, I'm not supposed to like legitimately publish it out on just throw it online anymore because Answers in Genesis wants to have copyright. So they have the copyright. I'm not able to just put it for free online because they want to have it in their own publications. So I took it off.
So there you go, Mr. Popular now, Praise. So, overall, what did we cover today? We had a lot of questions on abiogenesis, a lot of questions on testable predictions, address the strong indications that the model's working well, no more complaining. So there's a lot of there's definitely a lot for the critics to address. Dessel says, yeah, Aaron would be impossible. I would debate him tomorrow in a structured debate, like just if we could do like just 10-minute openings and then 3 to 5 minute rebuttals, it would be so fun to just, because Matt, the other day when in our video with Erica where she was teaching Aaron, it was right over his head on those molecular clocks. He said he didn't know Dr. Jensen. Like, he's really out of date. So imagine you just had timed rebuttals, how badly you would just annihilate him with the science.
Oh yeah, it was bad. When she was telling him about what Jensen did with a molecular clock, he goes, "Oh yeah, what I heard they did is they calibrated it completely wrong." He was talking about something entirely different. He didn't know what he was talking about. She was too embarrassed to even correct him. She was on his channel, right? So she's like, "Oh yeah."
Well, that's it. Yeah. Even she was probably thinking, I'm pretty sure you were in an after show with Dr. Jensen, but I mean, he is drinking a lot of beer during those, so he might have woken up the next day and completely forgot what he was doing. But you and I, Matt, were talking the other day about how they really do ignore the Y chromosome data because it's just such powerful evidence against their position, their story. And the successful predictions in the Y chromosome now represent one of the best evidences against deep time evolution and one of the best lines of evidence for our biblical creation position. And it just comes down to if the studies that we're talking about and referring to, the data we're talking about, Dr. Jensen's predictions and his work, if they all have such a glaring omission, why are they working so well? And they just don't want to address that. And when it comes to this dyad versus triad result, I pointed out in the comment section that they've already been performed for autosomal mutation rates. Okay, they're behind on the literature, and there's very little difference in the results between the two. You remember that, Matt?
Yeah, I mean, you can test any part of the mitochondria, the D loop, the individual hypervariable regions inside of it, the CO1 gene, or any of the 37 genes. It doesn't really matter. Dyad, triad, they all converge. They all show they're extremely fast. They can't slow the rate down no matter what they really try to do, except for when they merge it with the phylogenetic method, or when they include mutation rates that get 0 as their conclusion. They're trying everything they can, of course, but they still can't do it. It's amazing.
It is amazing. It is amazing. Well, we've gone another hour and a half. This has been fun. These critics got some explaining to do. So I think we're going to call it a night. It's been fun. Thank you so much. We got 20 people even at almost 5 a.m. So I'm going to go pass out. I've had a long day of reading through Y chromosome papers that I need a night off. But we'll be back at it tomorrow, won't we? Matt, you left me again, brother. I'm just kidding. I know you're with the kid. Okay, well, thank you so much, guys. Super stickers. Thanks so much, Doki Doki. Praise. Happy birthday, Dessel. Thanks for being here. Luca, Gavin, Taz. Nicholas, as always, makes some good points. He's a warrior in Christ. Oh, we had SpongeBob Imagination here. Yeah, great time. We'll be doing more soon. Dan's man says, "Oh, what? It's about to stop." I know, sorry. Should we pull an all-nighter? I don't think we can, Matt. We need some sleep. So thanks again, guys. God bless. It's been fun, and we will see you guys soon.